Hello everyone, my name is Charles Godfrey. I'm the director of the Oxford Martin School. Many of you know all about the Oxford Martin School. For those of you who don't, we're part of the university. We have a brief to do interdisciplinary work. We have a brief to do things that are linked with the major problems of the 21st century. And we're here because of the generosity of James Martin, Jim Martin, and the Martin family, and Jim's portrait is downstairs. So welcome to the latest in a series of lectures that we're doing this term, and it's an enormous pleasure to welcome Kevin Marsh, Professor Kevin Marsh, who'll be talking about prospects for new tools to control malaria. It's a particular pleasure because Kevin and I are old friends. Kevin worked for 25 years directing the Wellcome Trust Research Programme, a collaborative programme with KEMRI, the Kenyan Medical Research Institute, in Kilifi, north of Mombasa. And I had a mosquito project there, and I saw just what a fabulous community of Kenyan, UK, and actually global scientists that Kevin got together working there. Kevin has many interests around malaria and in other tropical diseases. Kevin is currently chair of the WHO Malaria Policy Advisory Committee. And to be honest, if I started reading out all the advisory boards that Kevin was involved in at the moment, there would be no time for his lecture, but it is hugely impressive. So with no more further ado, Kevin, can I invite you to talk on prospects for new tools to control malaria? Thanks, Charles.
Well, thanks very much, Charles, and thank you all for coming. I was sort of slightly overwhelmed when I walked in, saw how many people are here this evening. I was saying to Charles earlier, particularly nervous because one of the things I'm going to talk about does involve an area which Charles is a world expert on and I'm an absolute amateur in. So I've decided to turn this to a positive and sort of say when we get onto that bit, the questions that you'll no doubt have, I will refer all to Charles. What I want to do this evening is firstly, very briefly, give you a picture of where we're up to at the moment with malaria, the global situation, as a backdrop to ask the question, do we need new tools? Well, it's a slightly redundant question because as you'll have gathered from the title of the lecture, I obviously think we do need new tools. But think a little bit about what kind of tools we need and what kind of tools may be possible. And then towards the end, I want to bring us back to reflecting on the role, as it were, of technological approaches to things like controlling malaria, which I shall argue strongly for, but I want to sort of, at the very end, reflect on the things that we're missing if we only focus on technological solutions to problems like malaria. So firstly, the crash course for those of you who are not up to date with malaria, everything one needs to know about malaria. It's transmitted by the most dangerous animal in the world, the Anopheles mosquito, kill more people than any other animal. It's a parasite, which in that middle frame, you can see, tiny parasite, lives inside human red blood cells. And that's very important for one of the tools that I'm going to talk about later. And the reason we're bothered about it is it causes an enormous amount of disease and a really substantial number of deaths. So malaria is a global problem. It occurs throughout the tropics. There are two main human types of malaria. There are others. Plasmodium falciparum and Plasmodium vivax. They have slightly different distribution, as you can see there. Vivax is largely missing from Africa. And falciparum is by far the dominant in terms of disease burden and death burden. And you get a slightly skewed picture if we look at the world map, as we just did. If you take into account how many people there are and how many parasites there are, you get a rather different picture of the world. And from this, you can see that malaria is essentially a problem of Africa and South Asia. And that's because there are a lot of parasites in Africa and a lot of people in South Asia. Nonetheless, about 90% of all the deaths due to malaria occur in Africa. And for that reason, quite a lot of what I will say in the next few slides will refer to Africa, but it's generalizable to the rest of the world. But I'm going to focus on Africa, partly because that's where my interest lies, and partly because that's where, obviously, the major world problem is, as 90% of malaria deaths occur in Africa. If we go back less than 20 years, the world was very different. And sometimes I have to sort of pinch myself to realize that this is less than 20 years ago. Malaria was an absolute disaster. No one actually knew how many deaths there were. People guessed between 1 million and 3 million. I guess it was probably somewhere in between. It was largely, not entirely, due to the failure of anti-malarial drugs. We'd had a set of cheap, effective, relatively safe anti-malarial drugs which had just failed around the world. There seemed to be a lack of will to do anything about this, and yet it was recognized that malaria is a major impediment to economic development across many countries, particularly in Africa. And on the panel there, what you can see, just to talk you through that, is give some sort of indication of just how bad the situation was. So on the y-axis is the malaria-specific mortality, and then 3 blocks of time. One is pre-independence Africa, and it's important to recognize that at that time, there was essentially no malaria control in Africa, apart from in urban settings and some plantation settings. Essentially, malaria wasn't controlled. Post-independence, despite the many criticisms of African countries post-independence, you can see the situation did begin to change. But by the mid-1990s, the situation had had gone to a position which was actually worse than it had been pre-independence. So that's to say that towards the end of the last century, malaria was almost in a state of nature, as it were. It was essentially uncontrolled and responsible for an enormous number of episodes of illness and of deaths. So that was the situation less than 20 years ago. And at that time, eventually, the world was spurred to action, and I've listed some of the actions here, not all of them by any means. But they included the formation of new initiatives like Rollback Malaria, a whole set of public-private partnerships, which it will turn out are pretty important for what we're going to consider today. And then a whole set of well-meaning declarations in different cities around the world, the development of the Global Fund, and so on and so forth. And often, when these kind of actions happen, we are justifiably a little bit cynical about whether it will make a big difference. But in this case, I think there was a really remarkable difference. Within just a few years, global expenditure on malaria control had gone up by a log order. In 2004, it was round about just under $300 million a year, total global expenditure on malaria control. Pretty quickly, by 2009, 5 years later, it had gone up to $2 billion, and then subsequently rose a little bit more. So that was really a very major effect of concerted international action. Now, most of that funding, well, the funding, of course, goes on many different things, but essentially, malaria control is conceptually pretty straightforward. You either want to kill the parasites, or you want to kill the things which spread the parasites. And for killing the parasites, the traditional approach is drugs. It's important to note, I said earlier that one of the problems was that drugs had failed. And one of the big revolutions in malaria has been the development of so-called artemisinin combination therapies. This involves the combination of a very active, very fast-acting drug with another antimalarial drug, the idea being that each protects the other against the development of resistance. This was actually a major change. It's now the standard treatment across the world, and I'm very happy to say that colleagues in Oxford, particularly Nick White, played a very major role in the advocacy for the adoption of this approach, which is now a worldwide approach. And if you want to kill the vectors, there are many ways you can do that, and they're not all on this slide, but essentially, there are 2 major widespread approaches. One is the use of bed nets, which have been impregnated with an insecticide, which either repels or kills the mosquito when they land on it. And the other is spraying of houses internally with an insecticide, which again kills resting mosquitoes. So this is what malaria control, I mean, there are other elements to malaria control, of course. There are surveillance is incredibly important, and education, and so on and so forth. But essentially, in terms of actual tools, you either kill the parasite with drugs or you kill the vector with insecticides. Now, you would hope after that sort of rapid change of funding that there would have been a change in malaria, and in fact there has been. So on this slide, again, one doesn't need to worry too much about the kind of keys. I think everyone's used to squinting their eyes and looking at these kind of diagrams. Basically, red and yellow is bad, blue is better. And you can see that in just 15 years, this is measuring the amount, the number of parasites that were across the continent, that there's been a really dramatic change. I never pay too much attention to actual numbers when it comes to malaria because, to be honest, the surveillance mechanisms are so poor. So don't pay too much attention to the 600 million cases averted. But the trends, the percentage trends, are reasonably accurate. So mortality globally fell by 60%, and in Africa by closer to 70% in 15 years. And sometimes one hears this kind of thing, you think, well, is this just a result of the way you calculate the figures? And I have to say that, from our personal experience, as Charles mentioned, being based on the coast of Kenya, in a place where we began work because there was an enormous amount of malaria, over a period of 15 years, malaria didn't disappear, but it essentially became, or deaths due to malaria became a rare event. So certainly this general change was mirrored in people's individual experience. There was an absolutely dramatic change in just 15 years across Africa for malaria. So it all sounds as if it's going pretty well, but there are a few things to be concerned about. Firstly, although it was hoped that the combination of artemisinin with other drugs would give a breathing space and prevent the development of resistance, in fact, resistance is developing to artemisinins and to their partner drugs, particularly in the greater Mekong area. And this is important because historically, pretty much all antimalarial resistance to chloroquine and to SP and other drugs has begun in exactly the same part of the world. And in fact, it's a very precise part of the world. It's on the Thai-Cambodia border. We don't understand why that's been the case, but it is the case that that's where resistance always has begun. And a few years ago, it was noted that artemisinin resistance was beginning to emerge in this part of the world. Now, this is, at this stage, I think one has to be balanced about this. Many people have argued that this is terrifying, and it is terrifying. If this were to spread, this would effectively end our ability to treat malaria. But at the moment, it is relatively contained, as it were, in an area where there are not that many malaria episodes, which is a bit of an irony, that the problem is emerging where there is not that much malaria. Nonetheless, it would be very serious if this were to spread, so much so that in 2014, we recommended that the only approach to it was to go for attempting to eliminate malaria completely from the greater Mekong as a massive undertaking. The other cause for concern is the same sort of, if you like, evolutionary problem applied to insecticides. Of course, pathogens and vectors evolved to sort of avoid the effects of our attempts to control them. So again, on this map, not to get bogged down in the detail, but the browner it is, the worse it is, and the very dark brown means that there's resistance to all of the 4 classes of insecticide used for control of malaria vectors. On bed nets, there is only one class of insecticide which can be used because of safety issues. And then for indoor residual spraying, there are other classes. But essentially, as you glance at that picture, you can see that insecticide resistance is widespread, particularly in the very parts of the world where malaria is the biggest problem. And in many countries, now there is resistance to all classes of insecticide. So although things seem to be going well, there is great cause for concern that we have 2 essential approaches to controlling malaria: kill the parasites with drugs or kill the vectors with insecticides. And in both cases, there is resistance not simply emerging, but certainly in the case of insecticide resistance, very widespread already. So this is worrying. And this kind of a third cause for concern, which at first we might be tempted to say is related, but I'll try and sort of argue that it's not yet related, is that in the World Malaria Report last year, for the first time after several years of very optimistic reporting, the bottom line was progress has essentially stalled. And you see on the figure there, the red line is malaria deaths in Africa. When you dig down into the detail, essentially the figure this year is an estimated figure, and these are estimates, is really not different from last year. And in some countries, malaria deaths have begun to rise again. So after a period of optimism, this year, the bottom line was very different. The WHO message this year is that we're in danger of failing to meet all our international targets because the process is stalling. This is reflected to some degree. Now, you might say, well, that's obviously because of drug resistance and insecticide resistance. But in fact, the evidence to date is that that's not the case. Surprisingly, insecticide resistance does not yet seem to have impacted on deaths due to malaria, and neither has drug resistance to any perceptible degree. Much more likely what's happening is saturation and underfunding. By saturation, I mean saturation of effort. So on this slide are various measures of bed net ownership across Africa. And the one to really look at is the bottom blue line. which is the households which have enough impregnated bed nets for their requirements. Now, if you're an optimist, you could say this has gone up from, in fact, where it extended to the left, it started from a very low level. So you could say, well, this is great, it's gone up a lot. But we're still only at a level of covering around about 40% to the degree that we need to. So there's still a massive gap in applying the things that we know work. This is even more evident when you look at the uptake of antimalarial drugs. Again, each set of timelines it represents either treatment with any antimalarial or treatment with the antimalarial that you hope people will be treated with. And you can see two things. Firstly, that access to effective treatment is very low across Africa, even now, and more worryingly, it's not improving, and it hasn't improved for a long time. So we're in a situation where, despite all the investment, despite all the change, we're pretty much not making progress with access to treatment, and the progress we have made with access to treated bed nets is slowing right down. And I think there are some possible reasons for this. I showed you earlier the figure on the left, or part of the figure on the left, which shows the massive increase in international investment. But from 2010, the picture has been different. So from 2010, and the colors just represent where the investment comes from, but the bottom line of that is that funding has been flat. There's been no rise in funding. We're absolutely flat, and there's no sign that it will rise. And yet the amount of funding, around about 2.7 billion a year, is less than half of what we estimate it would take to apply the treatments and the tools we do have fully effectively. So we should not be surprised if progress is tailing off, even if our tools were working perfectly, which we've every indication that they will begin not to. There's another thing which I think we have to bear in mind. So on this figure is the proportion of malaria deaths in the world. And you can see that almost half of all the malaria deaths occur in 3 countries: Nigeria, DRC, and India. Now, if that's the case, what it means for Africa with Nigeria and DRC is that it doesn't matter how well individual countries may be doing, we're not going to see any perceptible fall further unless really high burden countries get on top of what is the most difficult problem. So I think there's no puzzle as to why things are plateauing. And it is probably not yet because of the failure of tools, but in the background, we also know that the tools are failing, so this situation is likely to get worse. So to summarize what we've said so far, there's been dramatic progress, genuinely dramatic progress in reducing the global malaria burden. But progress is in danger of stalling. Resistance to the main tools is building up in the background, and this is not optimistic for the future. So to answer the first question, clearly we need new tools. And I think there are 2 sorts of tools that we need. And I've called them standard tools and game-changing tools. And I feel a little bit bad about the standard tools because it implies that this is sort of business as usual, nothing too great about that. Whereas in fact, in many ways, what we urgently need right now are new standard tools. We need new drugs and we need new insecticides to replace the ones which are going to fail. So that area is in fact critically important. But I'm not going to say very much about it. I will say a little bit about it just to update you where we're up to in general with, at a high level, as it were, with that approach. And I would concentrate more on the idea of new tools, new paradigms that could really change the situation. But firstly, just to update on where we're up to with new insecticides. I mentioned earlier that one of the responses, one of the international responses to the dire situation at the end of the last century was the formation of public-private partnerships. Now, one of these formed a little bit later, actually, in 2005, was the Innovative Vector Control Consortium. And this brings together international funders, pharmaceutical companies, academia, to really tackle the idea that we need new insecticides. So people who want to know more about them can go to their website. But in brief, I think their greatest achievement has been they have facilitated the screening of 4 million compounds, and they have identified 9 new insecticide classes. And I think, although one should never be complacent about this, I think that it looks very good for the development of new insecticides. That's great. But of course, there's a lot between that and applying them. There's the cost, there's all the regulatory issues, and there's a time issue. we know insecticide resistance even now is widespread. So the question is not, will we have new insecticides? We will. The question is, will they be there in time, or will we have a great setback while we wait for them? Will they be affordable? But nonetheless, I think progress here has been very good, and we can expect it to be good. But will it be enough? Will it be fast enough? We don't know. Very much the same applies to drugs. So one of the earliest public-private partnerships set up was Medicines for Malaria. And this is very similar in approach to IVCC. That's to say, it brings together a whole lot of different stakeholders with a really targeted attempt to develop new anti-malarial drugs. Now, the next slide, you can't read, and I didn't expect anyone to read it. The point about this is it shows the portfolio, and across on the left in yellow are all the sort of, if you like, basic research promising leads, and across to the right are the things which are, as it were, already either in the field or moving towards the field. If you were able to read it, you would realize that all those towards the right are variations on a current theme. That doesn't mean they're bad ideas, they're great ideas. But all the genuinely new compounds, and there are some really exciting new compounds there, are way across to the left, which means there's going to be quite a gap. Although I don't doubt that some of these will come through and will come into practice and will have a big effect, there is still a substantial gap in terms of time and development before they can really answer what we need now. And at the bottom, I've put what's everyone's sort of grail, as it were, and this is called CRCAP, single exposure radical cure and prophylaxis. So this is the idea, which everyone agrees we want, but no one's quite sure how we're going to do, except that some of the drugs which are being developed look fairly promising. This is the idea that what you need is a drug which is safe, totally effective, with one dose, not only cures but prevents malaria. Of course, that'd be fantastic to have. But we're a long way from having that. But that's where this area of research is going. But one question which I think is important to ask, and lots of people have asked this, and I'm going to sort of just give you one example of the kind of answers which come up, is even if we had good tools, so if we had good insecticides and we used bed nets and we did spraying and we had good drugs, and even if we went to the extent of screening everybody in the population, finding all those who were positive and give them the drug, which seems a sensible thing to do, could you get rid of malaria? And I think the general consensus of all the attempts to look at this is no, you couldn't. In most areas, because of the robustness of the malaria transmission dynamics, even the best tools that we currently have, even if they did all work fine, and even if they were used to the degree we want them to. So what this, in brief, what this figure shows are 6 examples from across Africa. Only the one at the top left is in an area of low transmission. All the others are in varying degrees of relatively high transmission, which would be characteristic of most of Africa. And what the actual lines show are the effects over time, the modeled effects, of course, of introducing either bed nets alone, or bed nets plus indoor spraying, or bed nets plus spraying, plus screening, plus treating everybody in every possible combination. And the take-home message from this, and from other attempts to model this by other groups, is that except under lowish transmission conditions, even if you applied everything and it all worked, and you did it as well as you could reasonably do, you won't get rid of malaria. But, so what the index, as it were, is parasite prevalence in the community. But what you can see is there will be marked reductions in most settings, and that will be accompanied by a really marked reduction in clinical disease and death. So I'm in no way arguing that this is sort of not looking good. If we had good drugs, and we had enough funding, and good insecticides, and they were applied properly, we would have a massive effect on total load of malaria burden. But we couldn't eliminate it, is what most people believe, and what I believe, and what most modeling shows. So then the question is, if you wanted to eliminate and then eradicate, eliminate being something you do at a country level, eradication being a global result of all countries having eliminated, you would need new tools. And they would have to be tools which were conceptually different from the tools that we currently have and that we're currently developing. I'm going to talk about 2 approaches. The first is a pretty obvious one. It's what everyone says. Well, why don't you just develop a vaccine for malaria? And of course, it would be fantastic to have a vaccine for malaria. And as I'll indicate, as many of you know, I'm sure, we already kind of do. And the reason why everyone would love a vaccine for malaria is vaccines are the one public health intervention which across many different diseases have been able to leap over the restrictions of poor health systems, poor developmental settings. And the reason is because if you've got a good vaccine and it works and it lasts a long time, you can make contact with the population, protect, and sort of move away again. And this has been evident both with the smallpox eradication and even with polio. We haven't yet eradicated polio, but it's really remarkable how even across countries in the depths of sort of war and political disruption, it's been possible to maintain very high coverage of polio vaccination. So the beauty of vaccination is when you've got an effective, long-lasting vaccine, it can really shortcut a lot of the things which hold back more traditional approaches to health, as it were. So obviously, we want a malaria vaccine. And there is some encouragement. Humans do become immune to malaria. So this figure shows, I was going to say an imaginary population. It's not imaginary. It's a population we have done a lot of work with in Kenya. But it's, if you like, an idealized population in which on the y-axis is shown the maximum percentage of whatever it is you're measuring, and on the bottom is age. And what you can see is, over time, the green is severe malaria, for which you can read deaths due to malaria. And then the bluish is clinical episodes of a nasty febrile disease called malaria. And the pink is simply having malaria parasites, where you may not be ill at all, but you have the parasites. And what you can see is that over your lifetime, you do become immune in various senses to malaria. You become immune in a very important sense. You cease to be at risk of dying. And that's pretty good immunity. That's worth having. The problem is it's bought at the cost of a very high childhood mortality. So all the deaths are pushed down in those first few years of life. But eventually, you do become immune to dying from malaria. And if we could achieve that, we'd be very pleased. Of course, we want to achieve more with a vaccine than that. We would like to prevent transmission completely, prevent infection completely. But at least this gives an indication that it is conceptually possible to become immune to malaria, and therefore it's well worth thinking about vaccines. There are lots of reasons why it's been difficult. One of them, of course, is that malaria is a very complex organism. It has about 5,000 genes. It doesn't have the handful of genes that a virus has, or the sort of tens or hundreds that a bacterium has. It's a very complex animal. And developing a vaccine for malaria is more akin to developing a vaccine against cancer than it is to other infectious diseases. It has multiple stages. So shown there is the mosquito. It injects this sort of the wriggly-looking thing, the sporozoite, which finds its way to the liver. For those of you not familiar with that, that's a liver cell. It comes out into the blood, it invades again into red cells, and eventually it forms a stage which is transmitted by the mosquito. Each stage is different. Each stage has a completely different set of proteins and antigens. You can make an immune response to each stage, but it's going to be a very complex idea. And of course, the biggest problem is you can't take the traditional approach. The traditional approach to making good vaccines was to take whatever it was, mash it up, and inject it into people. And if you were lucky, it didn't do too much harm. And if you were very lucky, it gave a lot of protection. And the best vaccines, actually, were made like that. They're just mashing up without any knowledge of the mechanism. A mechanism has been worked out later. Now, you clearly can't do that with an organism which lives in human red cells. There's no way in which you could conceive of mashing up human tissue to produce a vaccine. Even if you could, it wouldn't actually work. We know from animal experiments that that approach simply doesn't work. So there are some real big issues. And this really then had to await for the development of alternative approaches to developing vaccines. And really the molecular era, as it were, where the idea is that you identify which bit of an organism it is which gives protection, and then you isolate that and you make your vaccine out of that. And work on malaria began with the sporozoite. That's the bit shown there, which the mosquito injects into the human. It just so happens that the first malarial gene cloned was the surface protein of the sporozoite. This wasn't totally by accident. I mean, there had been experiments showing that if you irradiate sporozoites, as opposed to mash them up and inject them into people, you can protect people. You can make them really quite immune. Not a practical approach, you would think, because, dissecting out the sporozoites from each individual mosquito, irradiating them and injecting them. But as a proof of principle, it showed that you could make a good response to this stage. And so for that reason, there was a lot of focus on this stage. And to cut a long story short, the major antigen from the surface was isolated. And in fact, the bit which is used in the current vaccine is just a repeating set of amino acids, a very, very simple motif which repeats. And that's stuck on to hepatitis B antigen shown there. And that is the current vaccine. So it's an extraordinarily small part of the whole malarial genome. And that, you would think, over-optimistic. You know, how can one squiddly little bit of just one of 5,000 proteins possibly do anything? Perhaps surprisingly, it does. So shown here is one of a number of studies which were done in the field showing, and the red line, so again, y-axis is number of episodes of malaria in a population of children who are vaccinated, and on the x-axis is time, and you can see those vaccinated with the RTSS, this is the putative malaria vaccine, versus those with rabies, that those with rabies vaccine as a control, those with the malaria vaccine did have quite a lot less malaria. And in fact, in this particular study, around about 50% protection. This was a study we were involved in. There are a number of these kind of studies. And on the basis of this, although 50% protection is not what you'd want, it was thought worth going to large-scale studies. And a large phase 3 trial of 16,000 children at 11 sites in Africa was carried out. And this produced an overall effect in young children of around about 36% protection against malarial disease. And I guess one of the questions there is, so is this a failure? I mean, we normally think of vaccines as being protective. We don't expect when you get a vaccine to still have the disease. So you could say, well, this is not a very good vaccine. But you could argue, I think, on two grounds that it's far from a failure. The first ground is that if you actually look at what would happen with this level of protection, because malaria is so frequent and so common and such a major cause of illness and death, well, you can read the slide for yourself, but essentially 200 children vaccinated would prevent more than 200 cases of malaria and 1 death. Now that ratio is better than pretty much any existing vaccine. So you could say it's a hopeless vaccine, but in terms of a public health effect, it will probably be better than pretty much any vaccine we've got. So in that sense, it's not a failure. I think there's another important sense in which it's not a failure, and that is we talked a few minutes ago about how conceptually difficult it is to make a vaccine against such a complex organism. And what this shows is that nonetheless, you can take one tiny bit of a very complex organism, and it can have a very significant effect. So if you're an optimist, and I definitely am an optimist about vaccination, you would argue that if one squiddly little bit of one of 5,000 proteins can produce 30% protection, there must be other squiddly bits in there that can do that. And if you add them all together, you'll get a good vaccine. So the challenge is, how do you find those squiddly little bits? And how do you spend sort of 1,000 years working through all the possibilities? And the time is a real issue. This is the RTSS timeline. Work began in the 80s. Clinical trials began in 2004, so 12 years before the phase 3 trials. This cost a couple of $100 million. And so the idea that we should go through every malarial protein and spend 30 years on every one of them, spending a couple of $100 million on every one of them, is clearly not tenable. And if you consider the sort of conceptually the malaria vaccine pipeline, there are in fact many potential targets sort of shown towards the left, as it were, under research. And then they get narrowed down, as things always do, as you go through various stages. But the absolutely crunch stage is the point where you need to go to a field trial, because we don't have a measure of what would protection look like. The only way to find out whether a putative vaccine actually works is to try it out. And this has now been done several times with other vaccines other than RTSS. Each trial involves, again, millions of dollars, many children, and a lot of time. And there are two major reasons why this isn't tenable. One is the cost, and the other is the ethics of it. That if you've done 3 or 4 of these and they don't work, you realize that what you're actually doing is performing experiments on children, not testing vaccines. And for all those reasons, it's clearly not possible to progress in that way. But fortunately, there have been a series of changes which I think sort of make a really big difference to what's going to happen in the future. The first is the genomics and bioinformatics revolution. Now, we could recapitulate everything we know about RTSS that took 30 years. We could recapitulate that in a sort of months, as it were. So you could now attach function to sequence. You can decide what your best candidates are. Perhaps one of the most important things is the move towards rapid down selection by controlled human malaria infection. For those of you not involved with this, it probably horrifies you, but the way that we propose doing this and the way we are doing it is you've got a candidate, you just give people malaria, and then you test the candidate. It means on a very small number of people, you can find out quickly whether it works. This has been done in America and the UK, here in Oxford, one of the major centres, for a long time. But the big change has been that a number of groups, including ours, and we were the first to do it, have now established this as an approach within Africa. It is actually, and of course you'd say he would say that, wouldn't he? But it is a remarkable, safe approach, a very effective way of screening down quickly as to what will work. So the combination of masses of bioinformatic information, rapid down selection, and then the third thing is a whole set of new ways of enhancing the immunogenicity of vaccines. And again, many places in different parts of the world are involved with this, but Oxford is a major centre for this with the Jenner Institute. So the combination of these three things, I think, mean that we can be really pretty confident that there will be another generation of malaria vaccines and that these will be combination vaccines. They'll take antigens, multiple antigens, from multiple stages of the parasite, and they'll be combined in a way which enhances their immunogenicity. And so if you'd have asked me 15 years ago what was the chance of a very effective malaria vaccine, I'd have said not very great. If you ask me now, I'd say pretty great, pretty good. I would expect, I think it will take some time, but I'd be very surprised if we can't produce a much, much better malaria vaccine. So I think that's very optimistic. There is a left field option here. You remember at the beginning, I said that the reason people focused on the sporozoite was that early experiments had shown that if you inject live sporozoites, not dead sporozoites, into people, you can make them immune. So there is one group led by Steve Hoffman who've taken this and driven it despite the rest of the world saying this is impossible. How can you make a vaccine where you have to dissect mosquitoes to produce the sporozoites? It's just all crazy. His group, Scenario, have actually really taken every problem and said it can be solved. So they're now doing mass industrialized robotic production of sporozoites. They are injecting them intravenously. Most people would say you can't have an intravenous vaccine, it's crazy. But producing in small numbers of people, high levels of protection. And there are, this year, trials will start in Kenya of this live vaccine. And there are plans quite quickly to move to large-scale trials in Bioko, Equatorial Guinea. So I think, you know, it's always good to have kind of controversy in a field. I think most of the malaria field is really pretty doubtful about this approach. And then there's a whole group of people who think, well, why not? It does work, and apparently you can solve the technical problem. So I think you will see a lot over the next few years about live malaria vaccines, which is a thing that you would have thought would not be feasible. So let me turn now to the second, if you like, potentially game-changing approach. When it comes to the mosquito, when I was being sort of taught about malaria, we were taught that the Anopheles mosquito, particularly Anopheles gambiae, which is the major vector in Africa, is a perfect transmission machine, that it's just so fantastic that you can't do much about it. So it was a bit of a shock, sort of, as I began to know not much about vectors, but a little bit from my colleagues, to realize that, in fact, that's not true, that even in very effective transmitting populations, a lot of the mosquitoes simply can't transmit malaria for a whole range of reasons. And when you think about it, that's not surprising. I mean, you know, transmitting malaria is not the mosquito's core business. That's not what it's been selected for. That's not what it does. It just happens to do it because it feeds on people. So it's not surprising that there is variation in the population, and some mosquitoes can and some can't. And as that's become more realized, of course, we've been able to work down to, well, what are the reasons for that? What are the genes which confer resistance? And that gives rise to the idea that what you'd really like to do is, as it were, have those genes take over in a population and render mosquitoes ineffective for transmitting malaria. Now, the problem with that is just ordinary, boring genetics. So many of you are familiar with this kind of diagram. I won't dwell on it, but basically, if the yellow gene is the one you want, this is the one you've identified as would confer resistance because of the way crossing takes over during mating, 50% of the progeny will carry it. And if that's the case, if you were to introduce this into a population, so say, right, we've got the gene we want, we put it into mosquitoes, we'll let all these mosquitoes into the population. By normal Mendelian inheritance, nothing would happen. The altered gene doesn't spread. It will either stay at the frequency it was put in at, or it'll by just chance disappear. Or if it's got some advantage, it might be by standard methods selected slowly over time. But you can't achieve what you want, is changing the population. So what you ideally want is something which is almost unbelievable. And certainly when I first came to sort of talk to people about this, I thought this cannot be true. And this is the bit where I'm going to look to Charles to sort of explain it afterwards if I fail to explain it. So if you go back to the idea that you've got a gene, the yellow gene, which you'd really like to spread quickly, imagine now that, go back to the bottom diagram, the cargo is the gene you want. If you could attach it to something, which when it comes alongside another chromosome, actually changes that chromosome to carry the same gene, then you get a very different thing. And in fact, this does happen in nature. As it turns out, most of you, I'm sure, have heard of CRISPR Cas9. This is a way, that's not the way it happens naturally in nature, but this works even better. So CRISPR Cas9 is essentially molecular scissors. You attach it to the gene you want, you target it to the site you want, it cuts the DNA, and DNA repairs itself. And when it repairs itself, what it does is copy what it's got in front of it. So it copies the very thing which caused the alteration. And so you end up with 2 copies of the gene you want to introduce to the population. And if you do that, you get the gene driven through the population. And the gene is always inherited. And this is the bit which I think is intuitively just completely surprising, and it still really surprised me. It doesn't matter if that gene is bad for the population. I mean, it will still be driven through. It doesn't matter if it causes the population to commit suicide. This is not a selective pressure. This is a mechanistic, must-happen thing, which, so long as the mechanism works, will happen. And that gives rise to 2 broad strategies. If you were to alter mosquito populations in this way, then you could either try and replace the population with the population you wanted, i.e., you've still got mosquitoes, but now they've got a gene which stops them transmitting malaria, or you could drive the population out. You could put in genes which are bad for the population and would get rid of the vector, which seems almost impossible. Does that actually happen? Well, it does. So I particularly like this. There are a number of studies now, but I particularly like this one from Anthony James's group, which took Anopheles steamersi, which is a mosquito that transmits malaria. It put in, I won't go into the details of it, but at the top you can see it did exactly this. It inserted, using the kind of mechanism I've talked about, 2 genes, the ones on the left, the orange and the purple. And these are genes which confer resistance to malaria infection. And then they did a really nice thing. I mean, it's irrelevant for the study, but it was very nice for sort of looking at how the results came out. They also put in a gene which made the eyes of the mosquito shine red if you put UV light on it. So I gave a very quick way of saying, have we really been able to push this gene into the mosquito population? Just shine a light at them, you get those nice red eyes. And the outcome was that, in fact, nearly 100% of the progeny actually did convert. They did exactly what you want. This is remarkably efficient in a laboratory setting. So this is the bit where I'm very nervous, because Charles is one of the authors of this next paper. The question comes up is, well, what would happen if you did that for real? Of course, a laboratory is a very unusual setting. In the real world, you've got all sorts of things. You've got population structure, you've got seasonality, you've got all sorts of things which will happen. But there's now some fairly sophisticated modeling approaches which allow you to at least attempt to say, well, what would happen? And this figure, which is complicated, but I'll try and sort of give an outline guide to it, is just one of the scenarios that would happen. And this is modeling the release of just 500 modified mosquitoes into a real population in a particular place in Tanzania. And if you look at the square, what that essentially shows is if you run these models many, many, many times, you can get various outcomes. And the outcomes you don't want are the ones in green dots, and that's your gene just disappears. So just to explain, again, along the x-axis, the homing rate is just a measure of how efficient your gene approaches, and the y-axis is a measure of fecundity. And given different assumptions, you could get different answers. You don't want the green dots. That's the gene disappears. It might be useful to have the black dots, where you get both genes persist to some degree in the population. It would depend to what degree that was. The yellow dots are where you actually lose the original mosquito, and now all your mosquitoes carry the gene, but it would depend on how many mosquitoes you actually had. And the red dots will be complete collapse of the mosquito population. And the remarkable thing, if you sort of move your eyes to the bottom panel, is the timing. That if you run these models, and they are models, of course, that in a remarkably fast time, within a couple of years, you could, by releasing, apparently, 500 altered mosquitoes, completely crash the population or completely replace the genes that you want to be taken out of the population. So, as I say, when I read this kind of stuff, I still have a sense of disbelief about it. But there's no reason to disbelieve it. This really could happen. And of course, it's produced great excitement. So much so that a lot of funding is being invested in this. This is the website of Target Malaria. This is, again, a consortium very heavily funded by Gates and others who are pushing this forward, working in Mali and Burkina Faso and Uganda. When I say working, I don't mean they're releasing modified mosquitoes, but they're doing the groundwork to do that. And there is real, I was going to say real drive, but that's because the thing is called gene drive. But there's a real enthusiasm that this could completely change the sort of transmission of malaria. But there are also some big issues. There are ecological concerns, ethical concerns, regulatory issues. And there's also biological, obviously. I said this was a mechanistic process. It is, but it is perfectly possible to evolve resistance to this process. So there are a whole set of issues. I don't want to give you the impression that next year you're going to see that malaria has been eradicated, but I do want to give the impression that this is an absolute paradigm shift and has sufficient behind it to make us at least believe that this could really happen. So it's certainly something important to know about. So finally, last couple of slides. We often say, well, I often say, a lot of people don't listen to this, that no country has ever eliminated malaria without a good health system. And I think it's important to realize because there's a lot of stress on elimination, as if it's just something where if you just try hard enough and you know, people make enough noise, you can do it. But I think it's really important to recognize that no country ever eliminated malaria without a good health system. Now, of course, a really good vaccine or gene drive could make that redundant. They're the two circumstances in which you could jump over the health system requirement. But there's a corollary, which I think is also really important for us to recognize. And that's that even now, no country with a well-organized, equitable health system has a significant malaria burden. Now, when you say this, people often say, oh, that's just that's a trick correlation. That's because all the countries with malaria are in a certain part of the world. They tend not to have good health systems. But in fact, I don't think that is the case. I think there are many examples which make this point, that a good health system, even with imperfect tools, even now, can make malaria a very minimal problem. And perhaps one good example, I kind of hesitate to say this because Haiti's had really bad press recently from President Trump, and I don't want to sort of contribute to this at all. But malaria was heavily endemic across the Caribbean. Cuba, which at an extraordinarily low GDP, eliminated malaria completely in the 1970s by having a very good public health system. Puerto Rico, which had a massive malaria problem at the beginning of the last century, eradicated it by 1962. But perhaps the best example is Hispaniola itself, where in a single island, you've got two countries, both with an equal population. The Dominican Republic has less than 10 malaria deaths a year, and they all occur on the border with Haiti. Haiti has 200 to 400 deaths a year. So this is a very clear example of the difference that a health system makes, even if you've got imperfect tools. I think it's very important that we remember that. So, final slide to summarize. I think there has been really extraordinary progress in reducing the global malaria burden in the last 15, 16, 17 years. But there is a danger that progress is stalling, it's plateaued. New drugs and insecticides are in the pipeline, but will they be there in time because the current ones are failing? Even with the standard tools and with good access, they'd have a massive effect in reducing malaria burden, but you probably can't eliminate in the places where you most want to eliminate. And to do that, we need new tools. There are two radical new approaches, both of which are substantially supported by experimental and theoretical evidence to suggest that they could work. We're not there yet, but I think one could be reasonably optimistic. However, despite all that, I think it would be very foolish to over-rely on the technological solution to what is essentially a developmental and economic problem, and to not realize that good health systems would actually obviate a lot of the need for new tools. So I hope that's given a reasonable balance between optimism and pessimism and sort of technological wizardry and kind of reality, as it were, and that's where I'd like to end. So thank you very much.
Kevin is very willing to take questions. We have some roving mics. Clara, we're recording this, aren't we?
We are, yes.
So just to remind people that we are recording it when you come up with odd jokes and things like that. Who would like to go first? Question there, chap in the blue.
Hello, thanks for the talk. I was wondering if you could comment on how a changing climate might change the regions with which the vectors at least can survive, and therefore malaria transmission.
Yeah, there's a lot of interest in this, obviously, and there are a lot of answers to it. It depends where you are. I mean, sort of UK-based malariologists are quite excited by the fact that we could end up with malaria back in the UK. So but I think it depends entirely on where you are. There's been a lot of modelling going on. There are some places where climate change would actually detrimentally affect malaria transmission, which will be good, although the other effects of it won't be good. And there are other places where you will get malaria transmission where you don't currently. And that's because transmission is fundamentally sensitive to temperature and rainfall. And climate change involves big changes in temperature and rainfall. So it could push it either way, depending on the circumstance.
Thanks for your talk, Kevin. My question is regarding the RTS vaccine trial. There was an increased rate in meningitis in the RTS arm. Is that a true rate, or is it that rabies has some non-specific protection?
Yeah. So this question refers to the fact that one of the outcomes, unexpected outcomes of the large trial I talked about of RTSS was the reporting of a much larger number of cases of meningitis in children in the vaccine arm. This was totally unexpected. And the honest answer is we don't know. There appears to be no temporal relationship at all to vaccination. There's, the meningitis was caused by multiple different pathogens, so there's nothing which links them all together. And there are a number of theories about what it is about. I don't think, well, I know most people don't believe it's mechanistically related to the vaccine, although there have been concerns about whether the adjuvants could in some way alter your response to other pathogens. And it also was far from even. So there were certain sites that accounted for the majority of those cases. Other sites, this wasn't the case. So it's a mystery, but it's one of the things which has been looked at very closely. What I didn't say is that following that trial, where you get 30% or so protection, the decision internationally was not to move to implementation, but to move to large-scale trial implementation in 3 countries in Africa. Because we need to know, like, will this have the public health effect which it could potentially have? But we also need to know, is that really an observation? I don't think anybody thinks it is, but you know, it's really important to know that. And then there are some other things which came out of the trial which clearly need to be sorted out. So that's why there's a kind of large-scale implementation rather than adoption of the vaccine.
You talk very eloquently about the importance of health systems, and there has been quite a lot of money going from donors into health systems, and an argument that this does some good but crowds out national health systems.
Yeah.
What's your view of the strategy of supporting health system development in Africa? An easy question.
Yeah, I think I've got a general view, which perhaps is not so helpful, which is that it's the same as everything, that all countries always solve in the end their own problems. That you know, this is true of education, this is true of health, this is true of all aspects of development, that in the end, no amount of well-meaning intervention by individuals, foundations, or other countries can really sort out the issues of any given country. It's a national obligation and requirement, and things only happen when it happens because of collective political will. So in that sense, I think you couldn't possibly put enough money in to change health systems in all the countries you need to change them. But I think what you can do is catalyse good practice. So I think you can focus. I'm not at all saying that external funding can't help. I think it can catalyze good practice. It can support research on what does work and what doesn't work, because it isn't that we know exactly how health systems should work. There's a lot of changes which can be made. So I think you can catalyze good practice, but in the end, and I think Cuba is a really good example. You know, whatever you thought of the political basis for it, Cuba solved all these problems. Cuba had a lower childhood mortality than the United States on a GDP of about $800 a year. So it's political will which makes the difference.
Other questions? Two here. Jack got his arm up fractionally first, and then just in front.
Thank you for the talk. I've got a question about the gene drive approach. Is that targeting a specific species of Anopheles gambiae, or is that a universal approach, considering that there is a different distribution of the abundancy of vector species, especially in Africa?
Yeah, well, I'll give you my answer, then Charles can give you the correct answer. No, initially it's species specific, and even within a species, of course, you may have population structure, which means that it's not completely interbreeding, so it's not as simple as it sounds, as it were. So what most people, I think, are thinking, that if you take a main transmitter like Anopheles gambiae, you can at least target Anopheles gambiae, but there are two things which may happen. Well, there's several things which may happen. Well, it may not work, but if it works, of course, there is population structure even in Gambia. We don't know that it was sort of spread like wildfire right the way through. There's also other populations could fill the niche vacated by Gambiae, which your genes can't move into. So maybe you just end up sort of replacing a problem with a problem. And also, of course, you could get the development of resistance. As I say, you know, it's a mechanistic issue, but of course, evolution is a powerful thing. And there will be things happening all the time in response to, well, in response is not quite right, but happening which could be selected for, which would confer an advantage on an Anopheles gambiae which wasn't susceptible. So I think, you know, it is targeted to an individual species, or even sometimes within a species to an interbreeding group. But Charles, you probably want to...
I mean, just very briefly, Kevin's exactly right. For anything to do with safety and regulations, we assume it will jump to anything that interbreeds. So within the Gambiae complex, Kevin's also right that the whole consortium spends a huge amount of time thinking about resistance, how you can avoid it using molecular strategies, and when it occurs, how you can have a long-term strategy to counteract it. And one of the excitements about the technique is, to a certain extent, it's a digital technique, so you can apply it several times. It's not like the case when you get Artemisian resistance, you have to develop a whole new system.
Yeah, thanks for the talk. My question is on the resistance starting at Southeast Asia. Do we know the underlying factors that are causing these, especially at the molecular level, so that whatever approaches we are using to combat the malaria, we could target it from there? Thank you.
Yeah, well, my answer to that is my standard answer to most questions, which is I don't know. And there's a lot of speculation about it. And you can imagine certain scenarios. So one speculation has been that there's something funny about parasites in Southeast Asia. And this has been seriously explored, the idea that you might have a hypermutating population. At the moment, I would say the evidence for that is not convincing. The other argument is that you've got a particular set of social circumstances, the widespread informal use of drugs, the use of drugs at low doses, fake drugs, and so on and so forth, which puts a big selective pressure. And the third is, if you like, an elaboration of that, which it may almost be like a founder effect. That's to say, because chloroquine resistance started there and spread around the world, then, of course, by the time it spread around the world, it had been long established in Southeast Asia. So Southeast Asia changes earlier than other countries to the next drug. So having been, by bad luck, the first place, you will almost automatically be the next place because you're so far ahead of the game. So I think those are the kind of things people are thinking about. There are particular social circumstances about migration and such like, which may increase the chances. But the honest answer is we don't know what's the main driver.
I'm afraid we're going to have to stop there because we have to finish at 6. Just before thanking Kevin again, let me tell you about the next 2 lectures in this series because they're both really interesting. One is called Antibiotics to Regulate or Not. which is by Steve Hoffman, who's visiting from York University, and Julian Savulesco from the Philosophy and Politics Department here. And that's next Wednesday, the 14th. And then the following day, they're coming thick and fast, we have one by Sarah Harper from the Growing and Aging, called The Growing and Aging Population, Implications and Solutions. Sarah's director of the Oxford Institute of Population Aging. And then we have an ad hoc event on the 27th called The Black Hole of Finance, Creation and Destruction in Life, the Economy, and the Universe. So do come along to that. I remember about 15 years ago, I went to another fabulous lecture Kevin gave, where you called Anopheles gambiae the most dangerous animal on Earth. So I started boasting to my students I worked on the most dangerous animal on Earth, and their derision when they found I didn't work on great white sharks or saltwater crocodiles, I had to stop it. Kevin, that was a fabulous talk, bringing your huge expertise in this area and a sort of combination of realism and optimism looking ahead. Thank you so much for talking to us.